All right. Good afternoon, everyone. Thanks for joining the coffee seminar series. My name is Eugene Rora, and I'm professor of the Center for Nutrition and Food Sciences, director of higher degree research training, or acting director, I should say. It is a pleasure to be with you today, and I would like to start by acknowledging country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. If I may have next slide, just a bit of a housekeeping reminders if you want. This seminar will not go beyond 1 p.m. So that's the usual time slot for the coffee seminar series, between 12 and 1 p.m. After the presentation, there will be an opportunity to ask questions to the speaker. I'll be managing the questions and answers. So please use that button on the bottom of Q&A. I would prefer not using the chat because it gets a bit confusing. Then let's, if possible, everyone using the Q&A button for this seminar series. So with any further to say, I would like to introduce our speaker today, Dr. Sharam Niknafs. Sharam, it's been a member of the coffee community for quite a few years already. He did his PhD at the CNFs between the 2015 and 2019, which means that I probably met him sometime in 2013 or 2014. So we've been collaborating together for quite a long time. It did take you 1 year or so to come from Iran. You could almost have walked, right? Sharam has been developing a fantastic program in CNFs, mostly related to poultry nutrition, but he's put his own spin into how to address issues, problems, hypotheses, particularly focusing a lot on mechanisms of action. He certainly was very important introducing in our group all the omics, transcriptomics, even proteomics, and that was, it's been ever since fingerprint of some of our activities in CNFs and coffee. So without further delay, I would like to introduce Sharam and his theme today, which is about gut functionality in chickens. Is that the key to sustainable poultry production? And that's what Sharam is going to tell us. So Sharam, the floor is yours.
All right. Thank you very much, Eugenie, for that nice introduction. I really appreciate that. I also do acknowledge the traditional owner of the land on which we meet. So I greatly appreciate the opportunity here by coffee, through which I can actually touch on some of the exciting projects that I have been involved during the last few years, basically. So the core of the presentation that I'm going to go through today is about poultry production, basically, and all of us are familiar with the amazing products that are coming out of poultry production systems, such as chicken meat and also, of course, eggs, and they are essential part of our food on daily basis, basically. So like every other production system, poultry production is also dealing with some challenges or some requirement or whatever we call it. So these challenges, I have just simply put them in some categories such as sustainability of the industry, animal welfare, and profitability. So we do have to take care of the environment in which we're basing our production system, and also, of course, the animal welfare side of the story, as well as the economic point of view. So And all these elements that I'm just talking about in the poultry production system, of course, it has so many different subcomponents, and they are all interconnected and very complex system, basically. So the real question is really, how do we go about this, and how are we going to address each and every one of these important elements in our industry? So if you're an animal scientist or animal nutritionist, the real question is, where do you want to put all your effort and all the research and all the investment to be able to contribute to contributing to improvement of these important areas in the industry, basically. So we believe that gastrointestinal tract or gut might be one of the most important candidates to be able to touch on each and every one of those areas. So gastrointestinal tract or gut is really a very interesting organ, if you can call it an organ. So it's kind of weird. It's inside the animal, and it's not inside the animal. It's like a tube that starts from the mouth and exits from the other side. But it has some really interesting properties that I'm going to take you through some of them. So if I ask you, for example, what's the main point of interaction of your body with the environment, you might answer skin, probably, but that's actually not the case, because when you look at the surface area in the skin and in gastrointestinal tract and you compare with them, you have average human being, you have a 2 square meter skin surface, whereas you have 16 times more of that surface within the gastrointestinal tract, and that's really amazing. So that's provides a very huge surface where the biological system is interacting with the surrounding environment. So, and also, of course, we've got the feed or the diet where it goes through this tube or the gut, basically. So look at that number, 70% of the production cost. So what that means is, if I'm a poultry producer and I'm a farmer, I put 70% of the money and investment that I'm making into the gastrointestinal tract of the chickens that I'm going to grow, basically. So that's a huge, huge number. So, and of course, in addition to that, nowadays, you're going to hear about microbiome everywhere. So there's literally tens of trillions of microorganisms that are residing in the gastrointestinal tract, and that's just a mind-blowing number. So it's way above the number of the cell that chicken itself has, basically. So these are just a few items that are really important when it comes to gastrointestinal tract or gut. So that's why gut functionality is such a key important parameter that you have to take into account if you really want to contribute to those parameters and those factors that I mentioned in my first slide. So of course, when it comes to gut functionality, you do have a couple of key important, the gut itself as a host, as well as the microbiome that is residing inside. And then, of course, you've got that powerful tool, which is the feed, that you can get the help from the feed and try to basically create an equilibrium between the gut and microbiome. And that's what we call it gut functionality. So the feed side of the story, it's really complex. So in the past, I mean, my mom used to just throw some whole grain wheat in front of the chickens, but that's no longer the case. So we have a feed that literally contains dozens of different nutrients and additives. So it's a really complex environment that we're talking about. It has probiotics, amino acid, prebiotic, enzymes, essential oils, and so on and so forth. So what I will be doing in the following sections of my presentation, I will be touching on some of these parameters. Of course, I cannot cover all of them. So in one of our projects, which was a small little nice project that we had, we were interested in looking at the effect of probiotic on the microbiome of the chickens, basically. So we all know that probiotics have a lot of good properties and characteristics that we can take advantage of to improve the gut functionality, especially when it comes to the microbiome side of the gut functionality, basically. So So in this project, as you see, when you actually supplementing the feed with different strains of lactobacillus, which are probiotics basically, and then you compare that to the negative control, which is a treatment 1 here, and then also the positive control, which is an antibiotic added to the feed, you do see that the probiotic that you have actually supplemented with the feed are increasing the diversity and the richness of the microbial community in the gastrointestinal tract. So they do have the same effect as the antibiotic when it comes to the richness. So what that tells us, that indicates that it might be actually useful to use those probiotic as an alternative for the antibiotic, because we do have a lot of pressure coming that antibiotics needs to be replaced with some other sustainable, profitable, and animal welfare friendly type of alternative. So probiotic is one of them. However, when you look at the other aspect is when you look at the antibiotic or treatment too, you see that even though it increases the diversity of the microbial community, but you're going to lose or you're going to change the abundance of some of the useful bacteria or microorganisms that are, for example, bacteroidetes. So that's they are contributing to, in fact, fermentation and production of short-chain fatty acids. So moving on to so that could be considered as a negative impact of the antibiotic. But when you compare that to one of the probiotic that we were testing in treatment 3, let's say, then what you see at the same time, you have an improvement in the diversity, but also it creates a uniformity across individual chickens. So that's a uniformity, what we call it uniformity of the microbial community. So the treatment-free probiotic that we have here, it's actually having the advantage of an antibiotic without having the bad, a negative impact of it on the abundance of some of the bacteria. So that could potentially be one of the alternatives for antibiotics. So that uniformity, coming back to that uniformity of microbial community, it's one of the contributors to having a uniform final product, and uniformity of the final product is absolutely crucial. It's a important indicator of how the production system is compatible with those parameters that I mentioned in my first slide, profitability and welfare, as well as the sustainability. So simply what it means, you have a population of chicken, and then they don't grow all the same way. So they're from the same genetic, the same environment, the same treatment, everything the same. You don't get the same, let's say, body weight. So that's, of course, a part of that relates to DNA and the variation on the DNA, but there is a significant part of it that relates to nutrition and nutritional requirements. So what happens, we're giving them a single diet or feed, and all of those chicken have to use that feed or that single diet. And then we are hypothesizing that basically the hypothesis was these slow-growing chicken might actually be facing some marginal deficiency if you're only providing them one single diet. You know, it's well balanced and good for some of the chickens and they do good. And for some others, they fall behind because we are not taking care of the nutritional requirement of them. So, but the real key question is, how do you want to find those key nutrients? How do you want to find that which nutrient is that is actually missing? So simply what we did was we had a bunch of slow grower chicken and fast grower chicken from the same population. And then you run a lot of systematic, what we call it two-choice test, so that you can identify what is missing in the feed, or what is more required in the feed of the chicken, whether it's a slow grower chicken or fast grower chicken. So what we found was quite interesting, because the slow growing chicken, when you provide them those two choices, what they significantly preferred non-essential amino acid that was added to the feed, basically. So, and that's quite cool, because when it comes to amino acid nutritional practices, what we do, or what is commonly being done in the industry is that the focus is here. Feed formulation is always done by essential amino acid, and they don't take into account for non-essential amino acids such as, let's say, alanine. So now, and the assumption here, of course, is because non-essential amino acids are the ones that are always being assumed that they are produced at a sufficient level within the body of the chicken, and that's why they don't take into account for them in the feed formulation. So, but what we have observed is that in the slow-growing chicken, in fact, they have that appetite, and they have shown that feeding behavior that they actually want those non-essential amino acids, particularly when it comes to alanine. So, and I'm putting focus here because I'm going to explain a bit more about alanine as well. So, of course, when we see that interesting feeding behavior and that appetite and that message that's coming from the chicken, hey, we need these, all right, then the first question is, okay, that might be related to the gastrointestinal tract. And the reason for that is, well, feed is going through the gastrointestinal tract, it gets digested, it gets absorbed, and also to a very high degree, it gets metabolized in the gastrointestinal tract. So, and it's interesting to know that the main source of energy in the gastrointestinal tract is also non-essential amino acids. So, of course, we throw there and we conducted a lot of analysis, I don't know, transcriptomics, proteomics, and even genomics, and as well as a lot of laboratory type of work. And what we have come up with is summarized in this. So this picture is basically a summarized version of 2-3 years of research in this project. So, so if you look at the alanine, when it goes through the gastrointestinal tract, it has to be sensed, and then it has to be transported into the cell, all right? But what we see in the slow-growing chicken, that is downregulated in slow-growing chicken, so it's not happening efficiently. But also alanine, as I mentioned, is being considered always as a non-essential amino acid, which means that you have this amino acid taken care of by the chicken itself. But apparently that's not the case, at least in the slow-growing chicken, because alanine has to be produced from the pyruvate, all right? And the production of pyruvate depends on the glucose uptake and glucose metabolism, which both are downregulated in slow-growing chicken. So you do have a deficiency in production of alanine, as well as deficiency in uptaking alanine from the feed. So on the other hand, you have an increased metabolism of alanine. And we believe this, in fact, is the mechanism behind that feeding behavior and that appetite that slow-growing chicken showed for alanine. So, and of course, we have published a part of this data in BMC Genomic recently. Just feel free to check out if you're interested. All right, so so far, probably you have noticed that my focus has been either microbiome or gut, all right? But right now we're having an active project. It's a very exciting project where we're trying to, in fact, tackle both of them at the same time. So we're using some tools such as Probiotic, for example, xylo oligosaccharide, and some enzymes, the most important one being xylanase. We're using these tools to, in fact, try to create that equilibrium between microbiome and gut at the same time and see whether we can do that or not. So in a very first experiment that we did in this project, we supplemented the feed simply with xylo oligosaccharide. And then what we saw was really interesting, because supplementing that level of xylo oligosaccharide, what gave us was a lower FCR. So for those of you that are not familiar with FCR, it's just feed conversion ratio. It's an indication how efficiently chicken are able to convert feed into muscle or meat, basically. So the lower the number, the better, basically. And then, of course, when you see something like that, a better feed efficiency by supplementing xylo oligosaccharide, then, well, you're going to ask yourself what happened in the gastrointestinal tract and what happened with the microbiome. So the gastrointestinal side of the story, when we analyzed what we found is that there's a bunch of biological system and metabolic pathways that are related to the immune system have been significantly enriched in the group that has been supplemented with xylo-oligosaccharide, all right? And then when you have a closer look at those biological pathways, you see, for example, all those letters that you see, each one of them indicates a gene or receptor or enzyme. So the green ones are upregulated, all right? So the ZOS in the gastrointestinal tract, in fact, is inducing and triggering the immune system components such as B cell and all those receptors that ends up higher, results into a higher production of IgA, which is a very important immune system factor to basically take care of the health of the gastrointestinal tract, as well as defending against pathogens that are enter into the gastrointestinal tract. So that's the gut side of the story. But as I mentioned, we're also interested to see what happened with the microbiome when you actually supplemented with the xylo-oligosaccharide. So these are upcoming experiments that we're going to have, and we will be looking at the effect of those on the microbiota, as well as what is the interaction with the enzyme when you're actually supplementing both into the feed. All right, so if you have noticed so far, the focus that I have put in here was more about what we call it post-hatching stage. So for example, in the case of broiler chicken, which is used for meat production, so we have a little chicken that you keep it for 42 days, and then during that 42 days, you can intervene and do all the intervention and nutritional practices that you want to actually achieve to improve gut functionality. So, but in addition to that, we do know that there's something that is called embryonic stage as well. So this embryonic stage, if you look at those numbers, is almost 1/3 of the chicken's life. So you have 21 days versus 42 days in the case of broiler chicken. So really, doing some intervention during the embryonic stage and complementing that with post-hatching period will actually maximize the gut functionality by those interventions that you're actually doing, all right? But what we will be doing, not only doing the interventions during embryonic stage, we go even beyond that, and we want to look at the maternal effect as well, because you've got these three important stages: maternal stage, embryonic stage, and post-hatching stage. Combining the intervention at all these stages together, we expect to see the maximized gut functionality in poultry production, basically. So, and then of course you need a tool to be able to do that. So the tool that we have selected to be able to look at all these stages and see how the complementary effect works across these three stages is essential oil. So they are just plant extracts, but they have a lot of volatile compound with very interesting properties such as antimicrobial, antioxidant, and of course they are stimulant of feed intake and digestion, which is relevant to the gastrointestinal tract, and also volatile transfers through biological tissues. That's important for us to, when we go across generations and look at the transition of those maternal effects. So this is the tool that we have selected for this project. Basically, going back to the embryonic stage. So the embryonic stage, you look at these pictures, there's a huge, huge complexity during embryonic stage. So you have 21 days, as I mentioned, you go from a single cell to a complete chicken. And it's changing so fast and rapidly. And all those components that you see on the left, they also change across all these 21 days. So it adds a lot of complexity to the work that you want to do, which is in ovo injection, which simply means delivering those substances, in our case, is essential oil, to the egg during those embryonic development. So when you want to do that, well, there's a lot of questions, fundamental questions that you have to answer. So as I mentioned, 21 days. Well, which day are you going to deliver these essential oils? Or for example, what is the compartment that you're targeting for delivering your substances, all right? And also you've got the pH of injection. During that 21 days of incubation, gas exchanges are happening and the pH is changing, and you're gonna add substance to it, and then you're making it even more complicated. So it's important to know all these to have all this information before even trying to intervene during the embryonic stage. So, and also, of course, you've got volume of injection. Well, clearly, we cannot inject 1 liter of essential oil in a single egg. So concentration of injection is also an important parameter. So to look at all these basic questions, we did run a lot of experiments, maybe 2 or 3 experiments for each one of those parameters. So to just, for example, pH of injection, volume of injection, within the range that we looked at, we didn't see any significant impact that they could have on the hatchability, which is an important parameter that we measure, basically. We do not want any negative impact on the hatchability when you want to actually intervene during embryonic development. It has commercial implication, it has animal welfare implications. That's not just going to be happening. So well, these 2 parameters, it seems that they don't have any impact. And then the same happens with site of injection, is the compartment where you're actually trying to deliver your essential oil. The same happened there. So we didn't see any impact. However, when it comes to day of injection, basically what we saw is it has to be strongly avoided injection at early embryonic development, such as day 0 or 4, or even maybe day 8. So because it's really detrimental for hatchability, and we're not going to be actually doing that. So, or concentration of injection, for example, I mean, it's probably a no-brainer. The higher the concentration, probably they get toxic, and then they will reduce the hatchability dramatically. So these are really valuable information that we have collected so far, all right? And then, as I mentioned, we're now entering into a very exciting stage of the project where we're trying to identify those really effective essential oil using those methods that we have established to look at how these essential oil affecting the gut functionality, whether it comes to on both sides of it, obviously, on the microbiome side of it, how well we can actually take advantage of those essential oil to establish a good microbiome right starting from the beginning of the development, as well as gut development in the embryo, basically. So take home messages. So so far what we've learned is that, okay, we do need to have a sustainable production system, profitable, and animal welfare has to be taken into account. And that's one important key that can actually help achieving that is gut functionality, which happens by creating really a good equilibrium between the gut itself and also microbiome within the gut, all right? So that's feed provides a very powerful tool. And when you when you combine those effects across different stages that I mentioned, maternal stage, embryonic stage, and also post-hatching stage, you do get, you do expect at least to see the maximized gut functionality in our production system, basically. So I think with that, I would like to thank the funding bodies, AgriFuture, Delacon, AB Vista, and Bioproton, that they actually allowed us to have fun with their money. So, and also, of course, I have a long list of friends and colleagues in Eugenie's group where without their help, obviously, none of these work could have been done. So particularly, I thank Asad, Mila, and Carla, who are 3 active PhD students in this project. So yeah, I think I'm pretty much done, and back to you, Eugenie.
Yes, thank you. Great presentation, Sharam. That's really a very good showcase of pretty much what 7 or 8 years of working in coffee, CNF. So well done indeed. So it was not easy to summarize so much into such a short time. So well done. Now, the session is open for questions and answer. Please use the question and answer button there to add any questions. While we're giving the audience some time to add questions, Sharam, I do have some icebreaker questions for you. So I was just looking at your plan of injecting in ovo essential oils, and I couldn't help thinking, well, how is that? Why do you expect that injecting essential oils will actually help at all in developing a more robust chicken? What is the rationale? Is that happening in the wild? Is that, you know, what's the rationale? Is there any rationale, or it's just a wild idea?
Well, as I mentioned, so the essential oils obviously have some very interesting properties, so antioxidant and antimicrobial and so on. So when you look at the commercial setup in hatcheries, let's say commercial hatcheries, so you may think that you have an ideal condition for chicken to develop in the hatchery, but that actually it's not probably a reality. So there's always a suboptimal conditions where you can actually take advantage of that injection of essential oil to reduce and mitigate those suboptimal conditions in the hatcheries. All right, so that's the first and immediate questions that benefit that comes to my mind. So, and also we do know that in ovo injection is somehow a commercial practice, is something that is already being done, especially when it comes to injecting vaccines or marek disease vaccines and so on. So we're trying to take advantage of that and supplement that marek disease with some of these essential oils to not only take advantage of the vaccine itself, but also complement it with all those interesting properties that I just mentioned in the presentation. So,
yeah, thanks so much. There are a few questions coming through. Just in addition to your comment, and I fully agree, but in the wild, I would expect that in the wild, chickens would have been used to eating a number of different plant extracts that will have some essential oils there and potentially being deposited in the egg. So it's not such a wild idea to think that essential oils may actually help in the development of the embryo, but that's, I don't know, just a wild guess. So we have a couple of questions. Thank you, John Dixon. Thanks, Sharon, for a great presentation. John is asking, what is the proportion of AMR associated with the use of antibiotics fed to chickens or other animals, if you know that, Sharon?
What proportion of AMR is associated with use of antibiotic feed?
So how important it is, the development of antimicrobial resistance as a consequence of antibiotics used in chickens, basically. I think that's the question, if I'm
right. So I think probably antimicrobial resistance is probably one of the main important factors when it comes to replacing and finding alternatives for it. So the industry wants to replace antibiotics, mainly because it creates resistance in the animal itself, which affects the health and welfare of the animal per se, and also there might be some residues left over that goes into human consumption as well. So what proportion exactly relates to antibiotic, I don't really have a number here, but I mean, if you use antibiotic, you do expect that there might be resistance. I mean, we have seen resistance, and some antibiotics nowadays don't even work, and they have been eliminated from the practices, basically.
Yes, there is a constant monitoring of the antimicrobial resistance in Australia. Australia is actually in a very good position compared to many other countries in the world, to be honest. So I think we're doing a good job in keeping those antimicrobial resistances down, at least when it comes to the use of antimicrobials in chickens and other species too. However, there's always a risk, and these antimicrobial resistant genes can be transferred from one type of bacteria to the next. And we do know that some of the antimicrobial resistant genes come from overseas through wild migrating birds and so on. So we always need to keep an eye on and monitoring this antimicrobial resistance. So it's a true and relevant issue in the poultry industry in Australia as well. We have another question from Marta. Thanks, Marta, for your question. She thinks you did a great presentation. How do you see the industry adopting your findings to reduce the variation in broiler growth?
So I mentioned about variation in growth in a couple of the projects when it comes to uniformity of the microbiome as well as the amino acid side of the story. So how do you adapt that in the finding? I think what I presented is obviously a proof of concept, basically. So we do find that, for example, using probiotic, all right, it increases the uniformity of the microbiome in the gut, all right? So in order to have a uniform final product, you do need to take care of all the factors that will contribute to that final product, and one of them is microbiota, basically. So if you have a uniform microbiota, that will contribute to have a uniform final product, all right? But when it comes to the amino acids, well, as I mentioned, so there's a lot of nutrients that are being discarded, not being discarded, but not being taken into account when it comes to feed formulation. All right. So how industry is willing to, in fact, whether adapting these into their practices or not, I'm not sure about that. Probably they will need a very strong proof for that, basically. So first, I think what I have presented is just a preliminary data and proof of concept. So if we do more work and if we prove that, yes, for example, supplementing feed with alanine or non-essential amino acid could actually increase the uniformity, that might be a good starting point for industry to actually think about it when it comes to feed formulation. And, you know, like everything else, like non-essential amino acid, the way that we do it for non-essential amino acid, we might actually be able to adapt the same strategy for non-essential amino acid as well. So if you formulate your feed, you always take care of, for example, lysine and methionine and all those amino acids. Well, I mean, I don't see a reason why you cannot do the same with non-essential amino acid as well.
Yeah, costs might be one, but as they get more popular, obviously costs will reduce. It is also probably worth stating here that there is already a move towards not alanine. Alanine, I think what you brought in your research about alanine is really novel and see where it goes. But there is an interesting relationship between alanine and glycine. And glycine, being a conditional essential amino acid in birds, is capturing more and more attention. And there's obviously a metabolic relationship between both glycine and alanine because they are linked. So there's something really already happening. And I think that the poultry industry and poultry nutritionists are paying more and more attention, at least to glycine. So it's maybe a bit early to speculate, but I think this is happening. So I think we'll see glycine probably appearing in some formulation sooner than later. Not yet, but we'll see that. One more question, Sharam, if I may. We keep talking about gut functionality. We talk about microbiome, and there's always somewhere behind the scene that we all talk about, and that's immune modulation and how the immune system is able to cope with all of these stresses, aggressions from the external environment. Now, in your concept of optimal gut functionality, what does the immune system play? What's the role of the immune system? Do we want to have a very sharp immune system, which could lead to inflammation, which obviously is negative, or you want to suppress the immune system? What do we want to do to avoid inflammatory type of processes? What are your thoughts about this?
Well, I mean, it's about the balance, I would assume. So you want an immune system that could actually be responsive when it has to be, and not being too sensitive when it doesn't have to be. So I think there are biomarkers that you can actually measure to how well responsive is immune system to the challenging environment that you're putting the chickens in. So I think the answer to the question, I guess, is just having a balanced immune system, basically. So to be responsive to the pathogens, but not being so sensitive that with every single challenge creates a inflammation, basically. And then we do know that the immune system will take a lot of energy. So having an immune system that is overactivated without any good reason, that will take a lot of energy and reduces the efficiency dramatically, especially in broiler chicken that is highly dependent on energy and protein as well. So
thanks so much. Look, there's another question that has not been put in the Q&A, but I know that Caroline Martin had a burning question to ask you. And what is it first, the chicken or the egg?
Well, for me, it's the egg, I guess, because I like eggs. So.
Is the egg because you like eggs? Okay, that's as good as anybody else's response. So, look, I think I don't see any other question, so we can probably go to the next slide to just present everyone with the next seminar. Please remember that the coffee seminar series happens every Tuesday from 12 to 1 p.m. And we are blessed with our next speaker on the 12th of July, to be honest, and that's about using natural abundance of nitrogen isotopes to identify more efficient cows in Northern Australia. And the speaker is Dr. Karen Eyre. So with that, I think that all, oh yes, this is, if you want additional information about this coffee science seminars, please see the links that are shown in screen. Craig Harner being the coordinator. Thank you, Craig, for your wonderful work in organizing this every week. Thanks so much. And with that, I'll leave you with a big thank you. And congratulations, Sharam. Well done. Thank you, everyone. Bye-bye.